Hello students, I am Professor Prafulla Kumar Mohanty, formerly Professor, Head and Dean Faculty of Science, Postgraduate Department of Zoology, Utkal University, Banivihar, Bhubaneswar. So today we will start another topic of Unit 1. That one is locomotion in protozoa. So this will be our today's topic. In fact, in last class we discussed about the different animals, different faunal diversity of our earth, and more particularly just in previous class we completed the different group of animal kingdom. Okay. So, one of the important groups of animal kingdoms is Protista, and today we will discuss a specific topic of Protistan group. Protista means mostly protozoans, and we will discuss how do these protozoans move. So, the topic of today's discussion will be locomotion in protozoa. Now, what sort of things we will discuss in this particular topic? What are the different aspects we have to cover, we have to complete under this particular topic? So, we will we will cover different segments which are like this. So, definition of locomotion, types of locomotion, locomotory organelles, structure of the locomotory organs, and functional dynamics. So, these aspects we have to cover under this particular topic, like locomotion in protozoa. So, when we think of locomotion, it is a very common term, but we have to define. When we are asked to define, sometimes it becomes very difficult on our part to define or to explain the things in simple language. So, what is locomotion? Locomotion is a term which has got its origin from the Latin term, from two different terms it has got its origin. The first segment of the term is locus. Locus means location, place, or maybe destination, and move here, and the second term is means to move. And motum, there is also another term which is known as motum. That is also another Latin term, the meaning of which is to move. Now, when we find the origin or etymology of the term locomotion, we will notice this term locomotion has got its origin from two different terms, from two different words, and these two words have been originated from the Latin term. The first segment is locus, L-O-C-U-S, the meaning of which is location or place or destination. So, locus means the place, and the second segment is movere, M-O-V-E-R-E, or motum, M-O-T-U-M, the meaning of which is to move, to progress, progression. So, locomotion is simply progression. So, if we define in simple language, we will find locomotion is defined as a process of movement of animals from one place to another. So, when animals do move from one place to another, that particular process of movement, that particular process of displacement from one place to another is called as locomotion. Therefore, I am defining once again for convenient remembering the term, locomotion is defined as a process of movement from one place to another through a particular medium. I repeat, locomotion is defined as a process of movement from one place to another through a particular medium. So, this is exactly the definition of movement. Now, when we talk of movement, you will also find one more peculiar feature in certain specific animals in the animal kingdom. That particular aspect is migration. So, you will find another term is migration. Migration means also the movement of animals, but try to differentiate and try to be specific in discovering the difference between locomotion and migration. Migration, the term has got its origin, migrare means also movement, particularly if you look at some specific fishes like hilsa, like eels, they do migrate for some specific purposes. Now, what is migration? Again, if you want to define the term migration, migration is defined as a periodic movement of animals from one place to another for some specific purpose, and those two specific purposes are feeding or breeding, or both feeding and breeding. So, some animals, particularly some specific groups of fishes, they do migrate from freshwater to freshwater, from freshwater to marine water or sea, or from sea to freshwater. So, to these different conditions of water, some specific fishes they do move, they migrate for fulfilling some specific purposes like breeding or feeding, or even both feeding and breeding. That is known as migration, and in fact, in fishes you will find various terms of migration like anadromous migration, catadromous migration, potamodromous migration, and oceanodromous migration. So, from different aquatic condition, from one aquatic condition, one aquatic environment, do animals migrate to move to another aquatic environment, another aquatic condition. That sort of particular movement specifically is termed as migration. Now, try to differentiate when you could know locomotion and migration, try to specifically pinpoint, try to answer and think of and ponder what exactly is the difference between locomotion and migration. So, migration of course, you can know, you can differentiate, you can establish the fact that migration is also a type of locomotion, but specifically it fulfills, it targets, it focuses on specific aspect of the life, either for breeding or for feeding or even for both feeding and breeding purpose. But if you look at locomotion of animals in general, and more specifically if you look at the process of locomotion in protozoans, you have to think of why do such microscopic organisms do move from one place to another. Yes, it has got some specific purpose of movement, specific purpose of displacement from one place to another. Now, what are the specific purposes? The specific purposes of these small creatures, small organisms of movement from one place to another are as follows. What are these? These purposes are: number one, it may be food, it may be shelter, it may be protection, and it may be what is called as a change of environment. So, these are certain specific purpose of the animals to move from one place to another. Now, what are the different purposes? The first one is the animals may be interested for taking food by changing their place from one place to another. This may also seek or need shelter to take rest, because that particular place where these animals are found, it may not be congenial or convenient to live there. So, it may be a shelter. It may also try, these animals may also try to protect themselves from a particular place, so that they do move. They change their particular destination place to another place, so that they can protect themselves. It may be protection, and when sometimes they do not feel that particular environment where they are existing or where they are available, animals are available, when they feel that particular environment is not congenial, is not convenient, is not comfortable, they do want to have a change. They change that particular place and they move to another place. It may be because of salinity, it may be because of pH, it may be alkalinity, it may be turbidity, it may be saline sort of things, or it may be hot, or it may be very cold. So, when they find some sort of, they want to have a change, when they need, when they seek, when they want for a particular change, they do change from one place to another. Finally, another segment and another important aspect of moving or movement from one place to another is partner. When the animals will want to have a partner, actually, I want a friend, let me change my particular place and discuss somewhere where my friends are available, or my mating partners are available. So, animals also do move for seeking, for taking the help of a partner, actually mating partner. So, these are 5 different purposes of the animals for the movement or for a change of a particular place from one place to another, change of a particular place. I repeat, actually what are the different purposes. Animals do move or they undertake locomotion either for food or for shelter or for protection or for change of environment or for seeking or for searching a partner. So, these are different purposes of locomotion, these are the different objectives of movement of animals from one place to another. So, now we completed definition, then we completed also the purposes along with that particular definition. Let us come to types of locomotion. Now, what are the different types of locomotion that you find? When you talk of locomotion, it is not of a singular type, it is of multiple type, and that multiple type of locomotion is based on different types of organelles found or existing on the animal body. Now, what are the different types of locomotion? Now, first one is creeping, this is also known as amoeboid. Second one is swimming, and third one is gliding. So, there are 3 different types of locomotion. The first one is creeping or amoeboid type of movement. Creeping means gurundi gurundi kiri jiba, creeping. When we were babies, actually we are creeping on the ground like a reptile, like a crocodile, like a turtle. So, that is known as creeping, gurundi gurundi jiba. Swimming, pohari pohari jiba, and gliding, आकाश को एमती फटकिनी बुड़ीकी गला भाया जीवा को ग्लाइडर जमती जाए, एरोप्लेन जम जाए, that particular process is called as gliding. Now, this creeping type of behavior, which is also known as amoeboid type of movement, it is found in amoeba actually, and particularly in a particular specific group you find you will find this type of movement, and this type of movement is found in Sarcodina. In which group do you find? Its group is Sarcodina. You find this particular type of behavior in Sarcodina, and you find swimming in ciliates and flagellates. Ciliates and flagellates, these are the groups, and this particular group is also known as Mastigophora group, and gliding is found in the group Sporozoa, which is in fact a parasitic group. Although apart from Sarcodina, you find in Mastigophores or in Sporozoa, you will also find in certain Mastigophorans and Sporozoans, you find creeping or amoeboid movement. But more commonly when you will come across this amoeboid movement is found in amoeba group of animals, and that is known as, that particular group is known as Sarcodina. We have already covered what exactly is Sarcodina, because it is a muscle type of body, does not have any external body organs for locomotion. So, Sarcodina, this is found, creeping type of movement or amoeboid type of movement is found in Sarcodina. The swimming type of movement is found in flagellates or ciliates, and gliding type of movement is found in Sporozoa, which is in fact a parasitic group. Now, let us come to one by one. When we come to creeping or amoeboid type of movement, it is achieved or fulfilled or facilitated by some specific organs. When we talk of movement, you do not find some specific organs available on the body in Sporozoan group. In Sporozoan groups, we do not find some specific organelles which can fulfill the purpose of movement. Whereas, when we look at amoeboid movement and swimming type of movement, some specific organs are present on the body of animals. Like for example, if you look at amoeboid movement, some specific organs in fact performs this particular movement, and these specific organs are known as pseudopodia. So, organ here is pseudopodia. The term pseudopodia has got its origin from the Greek term pseudes. Pseudes means in fact false, and I have already told you pos and podos means in fact foot. So, false foot. Please remember, as far as pseudopodia is concerned, the term pseudopodia has got its origin from the Greek word pseudes, not pseudo. It is not pseudo, it is wrong. The actual term is pseudes, P-S-E-U-D-A-S. Pseudes means false. So, these are the false foot, means temporary structures. They appear, they disappear. They appear immediately and they instantly also disappear, and these are finger like projections. So, these pseudopodia are finger like projections, actually blunt, finger like blunt projections. Okay? And when we talk of pseudopodia, the pseudopodia are of four types. Pseudopodium is the singular term, pseudopodium is singular, and pseudopodia are plural. Pseudopodia plural form. Please do remember. So, pseudopodia is, this particular term is a plural form, means number of pseudopodium. When it is more than one, it will be pseudopodia. When it will be single one, it will be pseudopodium. Now, there are four different types of pseudopodia which are found in this Sarcodina group. What are these? These four different pseudopodia are, the first one is Lobopodia. So, types of pseudopodia, the first one is Lobopodia, second one is Philopodia, third one is Rhizopodia, and fourth one is Actinopodia. So, this is known as the first one, first category of pseudopodia is Lobopodia, second one is Philopodia, third one is Rhizopodia. This is also known as, this Rhizopodia is also known as Reticulopodia, or this is also known as Mixopodia. This is also known as Mixopodia. And the fourth one is Actinopodia. This is also known, there is also another term for that one, that is also known as Axopodia. This is known as also Axopodia. Now, when you talk of this Lobopodia that is found in amoeba, you find this type of structure in amoeba. If this be amoeba, the structure that you find here, here, each one is a pseudopodium. Okay, it is finger like projection, it appears immediately, it also disappears. Okay. So, inside this particular animal you find two different type of fluid is there, liquid type of material is there. This one is, first part is known as plasma sol, and outer to plasma sol another fluid is there which is known as plasma gel. Plasma gel. For the first time, Libby Henrietta Hyman or L.H. Hyman explained the term, Libby Henrietta explained the term in Libby Henrietta in 1917 explained these activities of these two different types of fluids present inside this type of animal. When this fluid, in fact the interchange of the fluid inside the body takes place, the fluid comes out and the fluid goes in, fluid comes out, fluid goes in. So, this interchange of the fluid of plasma sol and plasma gel takes place inside the body of amoeba or amoeba like animals, so that pseudopodia are formed, the projections from the body are formed, and the animal gets convenience to move from one place to another by contraction and expansion of the body. So, this particular theory is known as sol-gel theory or viscosity theory. So, the sol-gel theory for the first time it was advocated by, put forth by, spoken by, described by Libby Henrietta Hyman in the year 1917, and this particular theory is known as sol-gel theory, or this is also known as viscosity theory, or this is known as viscosity theory, and this theory was put forth by L.H. Hyman in the year 1917. So, according to Hyman, this amoeba or this Lobopodia, they come to picture only when sol-gel movement takes place inside the body of the animal. There is interchange or flow of this two categories of fluid takes place inside the body that come to one side of the body. When this particular fluid comes to one side of the body, these projections, these finger like projections are formed. Okay. So, that exactly is known as Lobopodia. So, lobos means actually finger like projection that is found in amoeba, and they move to this sol-gel movement. Actually the animal gets chance to develop certain finger like projections, finger like structures, and it becomes convenient to move from one place to another. Then Philopodia, you will find Philopodia is like this, you will find the animal is like this. So, these animals, phylum means, in fact phylum means thread, this is the Latin term, and again pos and podos means foot. So, Philopodia means these are finger like projections, thread like structures actually. So, these are the thread like structures. These thread like structures in fact helps in movement of the animals from one place to another. That is exactly known as Philopodia. Third category is known as Reticulopodia. This is known as Reticulopodia or Rhizopodia. You will find Rhizopodia, rhizos means in fact root or root like structure. Here you will find the animal is like this, These are also projected structures, but these are fine filamentous structure. But the difference between Filopodia and Rhizopodia is these are somehow anastomos. The chonda chondi hekiri thanti. That is the reason why these are also known as reticulopodia. जेहेतु से माने छंदा छंदी थांती नेटवर्क बड़े थांती since these are available as like as net or nets, these pseudopodia are called as rhizopodia because it looks like they give an appearance exactly like root, they are exactly like net. Since these are root like, these are named as rhizopodia. Since these are like net, these are known as reticulopodia. Since these are slimy in nature actually, these are also known as mixopodia. So, rhizopodia, the third category of pseudopodia is rhizopodia or reticulopodia or mixopodia, and the fourth category is actinopodia. Now, what you find in actinopodia is you will find animal is like this and the projections they emerge, they come out like this. So, you will find these are the body structure, this is the body of the animal and this is the example of this particular type of things. This Filopodia is found in animals like Euglypha. I forgot to tell you, Lobopodia are found in amoeba or amoeba like animal. Filopodia are found in Euglypha or that related groups. In that particular related group you will find Filopodia, Rhizopodia or Reticulopodia or Mixopodia. These are found in animals like Foraminifera, Foraminiferans. One typical common example of one of the Foraminiferans is Elphidium, which is also known as Polystomela. So, that one is Elphidium or Polystomela. So, in Foraminiferans you will find, you will come across, you can visualize, you can observe Rhizopodia, whereas Actinopodia, what are the typical feature of Actinopodia? You will find Actinopodia, actis means ray. So, the Greek term actis means ray or spine actually. So, here when you find these structures, so these are Actinopodia actually. If you notice, if you observe these particular structures, these are very sharp spine like structure. So, these are also semi transparent in nature actually. These are semi transparent, these are all spine like actually, therefore, these are named as Actinopodia. And these Actinopodia are found in animals like Actinophrys, another animal is Actinosphaerium, so this type of structures are found in Actinophrys and Actinosphaerium. Look at one beautiful aspects actually. What you will find is these structures are found in from the name. Actinophrys means this spine like structure, sharp semi transparent spine like structures project out from the body exactly like this one. So, this is known as Actinosphaerium, whereas another animal is Actinophrys, same animal, same type, globular body like this, and from each circular structures you will find one spine like structures come out. This is from each segment you find one spine. So, that is the beauty, that is the structural characteristics or important feature of these four different categories of pseudopodia which are found in creeping or swimming type of animals. So, this part is complete actually. So, first part is complete. First category of movement is creeping or swimming, which is found in amoeba group of animals, that is performed, that is achieved by four different groups of pseudopodia. The first one is Lobopodia, second one is Filopodia, third one is Rhizopodia or Reticulopodia or Mixopodia, and fourth one is Actinopodia. Now, let us come to the second segment of movement. The second segment of movement is swimming type. So, what exactly is swimming type and which organs are responsible for the swimming type of movement? So, organelles or organs which are responsible to undertake to perform swimmings in animal movement of animals are known as motile organs or locomotory organs or locomotory organelles. So, let us see the second category of movement. So, the second category of movement in protozoa is known as swimming type of movement. Let me try to tell you one thing more emphatically. This first category of movement is found in the group Sarcodina, whereas swimming type of movement is found in ciliates and flagellates. Means in second category of movement you will find some specific critical complex structures are present in animals like cilia and flagella, and these cilia and flagella perform the swimming type of movement which are found in both ciliates and flagellates, and mostly this particular group is called as Ciliophora or Mastigophora. Mastix means, I have already told you, mastix means whip, Latin flagellum means whip, Latin cilium means eyelash. This thing I have already discussed in my earlier classes. These are the origin of different terms, origin and meaning of the terms which you can come across. So, mastix means whip, flagellum means whip, and cilium means eyelash. So, flagellum and cilium both are Latin, and the mastix is in fact the Greek term. So, these are the meaning of different terms that you find under the swimming head. So, under Mastigophora or Mastigophorans are characterized by swimming type of movement because of the presence of whip like structure which is known as flagellum. So, flagellum and cilium, they are morphologically and physiologically, these two organelles are identical. So, when you talk of the cilia and flagella, morphologically and physiologically they are identical, but there are differences, there are spectacular differences, significant differences between cilia and flagella or flagella and cilia. So, let me tell you one thing more. Actually, the branch of science which deals with the study of flagella and cilia or hair like structure is known as trichology. Is known as, it is either trichology or it is trichology. So, it is either trichology or trichology. So, trichology is the branch of science which deals with the study of cilia and flagella or hair like organs or organelles of the animals. So, this is known as trichology. Now, let us see the difference between cilia and flagella. What are the differences between cilia and flagella? What are the differences between cilia and flagella? Number 1, the number is 1 or 2, it is limited. Here number 1 you find the number is innumerable, the number is around 3000 to 14,000 or even more in each animal. So, if you look at this thing, here the number is restricted, it is restricted to 1 or 2 or maybe few. Here the number of cilia that you find in each animal, each organism, it is either 3000, it ranges from 3000 to 14,000 or even in certain cases it is much more than that. The second one, its size is 150 micron, whereas its size is 5 to 10 micron. And remember, 1 micron is 1,000th of a millimeter. So, 1 micron, I hope you know it, is equal to 10 to the power minus 3 millimeter. 1 micron, which is known as also mu. So, imagine the size or dimension of this particular structure. So, it is 150 micron, whereas The size of this one, it ranges from 5 to 10 micron. Number 3, this is normally found at one end of the body, whereas this is found throughout the body, surface. It is found throughout the body surface. Number 4, these structures are distributed in flagellates. These structures are found in flagellates, whereas these structures are found in ciliates. In certain larvae, the cilia are present, and flagella are also found. This is A. Flagella are also found in gametes. Flagella are also found in sperms. Flagella are also found in choanocytes of sponges. Choanocytes of the sponges. Okay. So, whereas ciliates are the cilia are present in ciliates, in certain larvae, and they are also found in certain tracts of the body, in certain body tracts, like for example respiratory tract, you also find cilia in certain body tracts. Then fifth point is, when you talk of flagellar movement and ciliary movement, the movement pattern is different. So, flagella normally shows undulation, whereas this exhibits a coordinated rhythmic movement. So, these are some major differences between flagella and cilia. Now, let me tell you once more how do you differentiate, or what are the differences between flagella and cilia. Flagellum is singular, flagella is plural term. Similarly, cilium is singular, cilia is plural. Please do remember. Now, let me tell you again what is the difference, what are the differences between flagella and cilia. Number 1, if you look at the number, it is restricted, very few, 1 or 2 or maybe few. Here the number is 3000 to 14,000 or more. The second point, the size of cilia 150 micron approximately, whereas that of cilia the size ranges from 5 to 10 micron approximately. If you look at the location or distribution of the structure on the body, this is restricted to only one side of the body, on the terminal part of the body. Like if you find this particular structure, you find this is found in one part of the body, whereas if you find cilia, these are found in the entire body. This is the difference. So, it is found in terminal part of the body, in one end of the body, that is flagellum is present in one end of the body, where cilia are distributed throughout the body. If you look at animal groups or animal body parts, where are these present? So, cilia are present in flagella are present in flagellates, flagella are present in gametes, flagella are present in sperms, flagella are also present in choanocytes of poriferans, choanocytes of sponges. Choanocyte is a type of cell which are found in poriferans, which are found in sponges. So, in choanocytes these flagella are present, whereas this cilia are present in ciliates, in ciliated epithelium, and in certain tracts like a respiratory tract, in certain reproductive tract of human body, even in mammalian body you find the presence of cilia, and also along with that you find cilia in certain larvae. So, these are the differences between flagella and cilia. Let us see what are the different types of cilia and what are the different types of flagella. Now, when we will discuss different types of cilia, you will find cilia are of two types. One is pterocilia, second one is kinocilia. This particular division or grouping is made on the basis of its functional dynamics. So, stereocilia means stereos means solid. This particular cilia, this cilia do not move, they do not almost move actually, immotile actually. So, these are immovable, whereas these are movable. These are normally found in ciliates, but stereocilia are found in macula. These are distributed in macula, internal ear. These are also found in another part which is known as epithelium of epididymis. Epithelium So, these are the differences of stereocilia and kinocilia. Now, let us see what are the different types of flagella. What are the different types of flagella? So, there are 5 types of flagella. The first type of flagella that you find is known as stichonematic. Stichonematic. Second one is pantonematic. Third category of flagella is known as acronematic, and fourth type of flagella is known as pentachronematic, and fifth category is simple type. So, these are 5 categories of flagella you can come across as far as structure or structural architecture or structural configuration is concerned. You will notice 5 different types of flagella. So, I repeat actually, the first category of flagella that you find is stichonematic. The second category of flagella is known as pantonematic. Third category of flagella are known as acronematic. Fourth category of flagella is known as pentachronematic, and fifth one is very simple one where you do not find these myonemes. Now, when you talk of flagella actually, there must be the presence of thread like structure actually, they help in this particular movement. Now, let us see how can we differentiate as far as the structure or structural architecture is concerned, how can we visualize, how can we differentiate. Let us draw the diagram and let us try to differentiate between all these. When we talk of the stichonematic, you will find the structure is like this. So, this is stichonematic. In one side of the body you find this thread like structure. In pantonematic you will find like this. In acronematic, pantonematic means in both the sides you find this fibrillar structure, and in acronematic what you will find? You will find like this. In one end you will find this pentachromatic, you will find pentachromatic, you will find both the sides or in multiple sides along with the terminal part you find, and in simple type it is very simple, I mean it is not very critical actually. So, let me tell you again, as far as diagrams are concerned, these are illustrated, these diagrams are self explanatory. As far as stichonematic type of flagellum is concerned, you will find a single sort of structure or axis you find in one side you find this type of structures, fibrillar structure. In pantonymatic you find both the sides, and in acronymatic you find only on the terminal part. Acros means end or terminal part or terminus. In end part you find this fibrillar structure. In pentachromatic you find in more than two sides and the terminal part the presence of these fibrils, and in simple type it is almost absent actually. So, in toto you find, as far as this different varieties, different types of flagella are concerned, you find 5 different types. So, stichonomatic, pantonymatic, acronymatic, pentachromatic and also simple type. Okay. So, this is in fact the different types of flagella. Let us see the structure, microscopic ultrastructure of flagellum or cilia. Now, this is the ultrastructure of the flagellum. When you will take a vertical section of a flagellum, this particular picture we will get. Now, what structures do we find inside this particular flagellum? These are very microscopic. When we see at this flagellum, they are very small actually. I told almost 150 micron in size, but if you take a vertical section or a longitudinal section, the internal structure will be like this. What we will find? We will find this is the tip of the flagellum. This entire flagellum is covered by a membrane which is known as plasma membrane, and in the center of this particular structure you will find 2 fibrils are there at the center. So, these are 2 central fibrils, and in the periphery these 2 central fibrils are surrounded by 9 peripheral fibrils. These are 9 peripheral fibrils, and these are this is the basal plate, this is the root knob and the rootlet, and this rootlet or these structures are embedded inside the body, and this is projected out. This part is embedded inside the body and this part is projected out, which enables the animal, which helps the animal for movement. So, this is the ultrastructure of this particular flagellum. These flagella or these structures actually they contain some fibrillar structure actually. These are also known as this both flagella and cilia, also known as microtubules or microtubular structure actually, and furthermore they contain some thread like structure which are known as myonemes, which are also known as these myonemes. These myonemes they help in movement of the animals actually. Okay. Myonemes, myo means muscle, nematos means thread. These muscles are almost thread like structures, and these thread like structures helps the animal for propagation, for progression, for progression or for movement or for displacement from one place to another. Then what is the role, the functional part? The last part is the structure of the locomotory organ and last part is the functional dynamics. What do they do? That we have to see. When the animals they try to move or they think of a displacement, what do they do? We can take an example. Supposing this is an animal. This starts movement actually. This is an animal actually, this is an organism, this is one of the protozoans. They start to move, and this is the example of a protozoa which is monas by name. This is the example of monas. It is a protozoa actually. When this particular animal wants to move, what happens? This starts movement actually, this is a question of undulation. It starts moving like this, and when it stops movement actually, that means this part becomes stable and only the apical part becomes in vibrating structure. So, after this one it becomes very straight like this, and then it moves. When it tries to move, then it starts undulation actually. So, always when the animal tries to move, it moves like this, like a serpentine movement it tries to move, and gradually it stops the basal part becomes straight and the apical part becomes wave like structure. So, like this actually undulation, through undulating movement the animal moves, then it becomes stress, then undulation starts, like that actually the progression of the animal takes place. In some other animals like Paranemia, there is another animal which is known as Paranemia. You find how do animals move. Supposing this is the animal, so this particular stroke is known as effective stroke. The animal starts movement means this is known as effective stroke. That means when this animal, this is another protozoa which is known as Paranemia, when the animal tries to move through this particular flagella sort of thing, it starts movement like this. This is first, second, third, fourth, fifth, sixth, like this. So, this is the first position, this is the original position of the flagellum, then it occupies this position, then like this, then like this, then like this, then it comes back. When it moves like this means it grows ahead, goes forward actually. Then when it goes reverse actually, the animal moves like this actually. When it goes back, what will happen? It becomes like this. This is the original position, this will be 7, this will be 8, like this, 7, 8, 9, 10, 11, it comes to the static position. Then when it comes to this position, again it will come to this position, that means this comes to this position, then finally it comes to this position. Likewise, this position and this position, this is known as effective stroke, whereas this particular activity is known as what exactly is called as recovery stroke. This is known as recovery stroke. So, this is known as effective stroke of movement, this is known as recovery stroke of movement, means initiation when the animal starts movement, it gets initiated from this point, come to this position, and it once recovery means this recovery from this point, it comes to this position. So, one complete phase of movement is over. So, when it tries to do like this, it goes forward, when it comes to original position like this, it remains like this, then it tries to do like this, then it moves forward. Likewise, the animal tries to move from one place to another. Then finally, so this is regarding this swimming type of movement which is found in ciliates or flagellates, and the last group is gliding type of movement. Here this is found, this particular type of movement is found, or gliding type of movement is found in a specific group of protozoa or protist, that is porozoa, which is a parasitic one. Now, what is unique to this particular group of organism is, in this group you do not find, you will not notice, you will never come across some specific locomotory organelles or motile organs on the body. So, the body surface, in no way you will notice the existence or availability of specific motile organs or locomotory organs. It is simply a spore like structure and it simply moves by a particular technique which is known as gliding. So, with this we complete the movements, how do animals, how do protozoans move. Let me summarize by telling like this, what did we discuss or complete in this particular session. So, the topic of deliberation or discussion of today's class or today's session was locomotion in protozoa. Now, what sort of branches under this one, under locomotion of protozoa, what did we discuss? First we started with the definition, what is the definition of locomotion, then types of locomotion, then locomotory organs, what are the different locomotory organs present on the animal body, the structural details of the locomotory organs, both this pseudopodia and flagella and cilia and the functional dynamics. When we find the locomotory organs are existing on the body, what sort of activities do they perform, so that animal gets opportunity or scope to move from one place to another. I hope you must have remembered all this. Please read out the things in detail so that you can have better conceptual clarity. Thank you.